Right, so welcome back to our next year 13 lesson on photosynthesis, the light dependent reaction. One of my favourites, this one. I'm not going to lie to you, this is probably going to be initially a bit of a flyby. It's going to be difficult for you. I'm not going to lie, but it's going to take some effort on your part to really go through it. Look at those notes I've given you. Maybe watch this video a couple of times, make your own notes to really kind of get your head around it. Once you do get your head around it, and you will, everybody does get there with it eventually, it will be a really invaluable topic to have in your arsenal because it's one that you can pepper into a lot of essays. Photosynthesis you can usually shoehorn into many things because it's such a fundamental process. And this one is challenging, I'm not going to lie to you. There's a lot of moving parts, there's a lot of complexity, there's a lot of vocabulary, a lot of new key words for you to get your head around. So it's going to take time for you guys to process that, but you need to work on it yourselves. You need to really get to grips with it. So the light dependent reaction depends on light. It uses light energy. So the first part is that from the last video, that kind of capturing of the light energy with the use of chlorophyll, and I'm going to talk quite a bit about chlorophyll in a moment. And then there's a series of key kind of events that's going on. We've got what's called the electron transport chain. We've got ATP synthase and this process called chemoosmosis that's going on. And we've also got another process called photolysis or photolysis, however you want to pronounce it. So there's a lot of very big new words, and don't worry, I'll go through each of those key processes with a diagram I've produced on PowerPoint that hopefully will help you understand it a bit better. But before we get cracking and really start looking at these kind of key components, there's a couple of important chemistry terms that you need to be aware of, and those are oxidation and reduction. Okay, so hopefully you remember this from GCSE chemistry and you have a bit of an idea, but we can all do with a warm-up. And the best way I have remembering this is a mnemonic called oil rig. Okay, so oxidation is loss, reduction is gain. Okay, and now that applies to electrons and hydrogen atoms, but you get the opposite with oxygen, which makes things a little bit more complicated. Generally speaking, in the biology, we're only ever really looking at electrons and hydrogen, but bear that in mind. So something for oxidation is the loss of electrons and hydrogen. That E minus there is a symbol for electrons, and reduction is gain of electrons and hydrogen. But again, bear in mind, oxidation is the gain of oxygen and reduction is the loss of oxygen. But we're going to see this idea of oxidation and reduction a fair bit in photosynthesis and in respiration. So you've got to start getting your head around it. So if something gains electrons, it will become reduced. If it loses electrons, it will become oxidized. If something gains hydrogen atoms, it becomes reduced. If it loses hydrogen atoms, it becomes reduced. And I've actually got a couple of sentences for you guys to complete on the notes to see if you've got that idea of oxidation and reduction. Okay, so without further ado, let's move on and look at the role of chlorophyll and photosystems in the light-dependent reaction. Right then, so chlorophyll is a really interesting molecule, and it actually has got magnesium in it, so it's like a sort of prosthetic group, similar to kind of hemoglobin has iron in it. And that magnesium is really, really important, but we'll get back to that in a minute. So chlorophyll is considered to be the primary photosynthetic pigment. We've got chlorophyll A and chlorophyll B, which are able to absorb slightly different wavelengths of visible light. But there are other what we call accessory pigments like carotenoids and xanthophylls. And these we don't normally see them because when we see a leaf, we see the green colour, and that is the chlorophyll that's visible. But when leaves go through autumn, they will break down and absorb the components of chlorophyll for next year. But then you might start seeing those autumn colours, oranges and reds. And they're the accessory pigments which are now visible, like carotenoids and xanthophylls, that have always been there, but they're now available to see. Now, they're not just randomly embedded within the thylakoid membranes, they have some degree of structure. And what I've drawn here is a very kind of simplified diagram of a photosystem. Okay? So at the base of the photosystem is our primary photosynthetic pigment, in this case chlorophyll. And then we've got other accessory pigments there. And what happens is if a photon, light, hits this structure, if it hits the chlorophyll dead on, perfect. If it hits these accessory pigments, they will channel that light down or pass that energy down to the chlorophyll. Okay? So once the chlorophyll has absorbed light energy, it excites, this is the way to word it, excites a pair of electrons within it. They become excited, they go to a higher energy level. And when they do that, they will leave the chlorophyll molecule. Okay? And as a result, because the chlorophyll has lost electrons, it has become oxidized. And actually this process here, another key word for this, where it absorbs light and loses electron, another term you could use here is photoionization, because remember, ionization is all about loss or gain of electrons. So the chlorophyll loses electrons, those excited electrons, but where do they go? Well, they go on to the next phase of this process, and this is probably where we're going to start cutting to that diagram so I can talk you through it. So we've got the next phase that we go through is what we call the electron transport chain, and this is where we've got a series of electron carriers. These are proteins embedded within the thylakoid membrane, and the electrons are passed between them. Now, as they're passed, as say the first acceptor gains electron, it becomes reduced. When it passes it to the next, it becomes oxidized, and the next one becomes reduced. So we call this a series of kind of redox reactions, reduction, oxidation, over and over again. And as the electrons are passed along the chain, they transfer energy, and they go down to a lower energy state as they go along. This energy is used in part to generate some ATP, okay, and it's also used to help pump protons, H plus ions, however you want to word them, from the stroma, okay, into this what we call the thylakoid space. So within the each thylakoid is like a little disc with a membrane all the way around it, and inside is this sort of impermeable layer, or impermeable space. So the H plus ions cannot cross the phospholipid bilayer because they are charged. So the only way they can get in or out of these thylakoid membranes is through specialized proteins. Okay? In this case, they are being pumped into that thylakoid space using the energy from those electrons. Okay? So that's an important part, because ATP, being the product there of the electron transport chain, is one of the key products of the light-dependent reaction. So what happens to those electrons? I'll talk about their fate later on. What happens to those protons? Well, let's have a look at them next. So now I've started to pump in some protons within my thylakoid space. There's another process that actually generates even more protons, even more H plus ions. And that process is called photolysis. So that's splitting water with light, because photo means light, lysis means splitting. So below the photosystem, photolysis would occur, and the light energy splits the water into, well, the hydrogen is split into protons and electrons, and then oxygen is also released. And that's just where that, if we think about the photosynthesis equation, that is where oxygen comes into that equation, is right here because of photolysis in the light-dependent reaction. So the electrons that are released, they actually replace the ones that have been lost from chlorophyll. And that way, the chlorophyll molecule can then carry on absorbing light energy. It kind of regenerates the chlorophyll, so it can carry on becoming, those electrons can now become excited, they can go on the ETC and so forth, and another round of photolysis would replace them and so forth. So we've now got a very, very high concentration of H plus ions within our thylakoid space. Some that have been generated through photolysis, others that have been pumped in by the ETC. Now, we've now developed this very, very high concentration, and we've got a low concentration in the stroma. So they want to diffuse out, but because they're charged ions, we know that they cannot simply diffuse out. They need a channel, they need a way to get out, they need some sort of facilitated diffusion. And the only way out is through an enzyme, and that enzyme is called ATP synthase, one of the coolest enzymes in the universe, if you ask me. And it really is the power station of our cells. It's where the vast majority of ATP is synthesized. Now, it's located in chloroplasts. It's also located in mitochondria. Same exact enzyme. So it's a very, very important kind of universal enzyme. So we've now generated this very kind of steep concentration gradient, a high concentration within the thylakoid space and a low concentration within the stroma. So those protons begin to flow out into the stroma through that channel, the ATP synthase. And we call that chemiosmosis or chemiosmosis theory. And as those H+ ions are flowing through the ATP synthase, they are actually causing that headpiece of that enzyme to turn round and round. Actually, I think of it as a bit like a turbine. You know, how wind or water or steam might turn a turbine generate electricity. Here the protons are causing that ATP synthase headpiece to start to rotate, and that will cause its active site to change shape, and what that will do is phosphorylate ADP and phosphate into ATP. So here is where we're transferring a lot of that energy into those high energy chemical bonds in ATP. So that, don't forget to include that keyword of chemiosmosis when you're describing how the protons are flowing out and referring to the concentration gradients and so forth. So those H+ ions are flooded back out into the stroma. They've done their work. Some of them are going to actually rejoin with those low energy electrons that have come off the ETC, the electron transport chain, and they're going to reform hydrogen atoms again. And what is going to come along now is something called a coenzyme. So these aren't actually true enzymes, but they do aid enzyme action. And this one is called NADP. Okay, NADP. And it's an electron acceptor, hydrogen acceptor. And what it does is it will transport, it will take on board some of those hydrogen atoms. And in doing so, because it's gained hydrogen, it has become reduced. Okay? And that is the other key product of this light-dependent reaction. So we'll summarise some of those products that we've seen from that very complex light-dependent reaction. Cool. After that lot, I need a cheeky tea break, I reckon. Gorgeous. So I might have made it seem a little bit effortless. I've done it a lot of times, many, many years now. So I've kind of learnt it. But you guys can too. And I suggest that maybe you pause it here, re-watch it again, make notes as you're watching it again, use the notes I've given you to really kind of start to build up your knowledge, and hopefully you eventually will become fluent and you'll know how to word it specifically. And I think once we start practicing some exam questions too, you will pick up on it. So let's recap, let's summarise, and let's look at the key products of this light dependent reaction. So product 1 is ATP. Product 2 is reduced NADP. You can write it like that, or if you like me, I'm sometimes a bit lazy, you can write it like that, because it means it means the same thing. And of course, we've also produced some oxygen. So I'll start with the oxygen because in some ways it's the least important. It's simply a byproduct of photolysis, but it's become incredibly important for all aerobic life forms on the planet, including myself right now. Some of that oxygen will be used in respiration, and some of it will diffuse out of the leaf. And if you were to measure oxygen uptake, oxygen release as a measure of photosynthesis, bear in mind it wouldn't be 100% precise, because some of that oxygen is used by the plant itself for respiration. These two, ATP and reduced NADP, well, these are both going to be now used for the next stage of photosynthesis, what we call the light independent reaction, okay, which is that metabolic pathway we call the Calvin cycle. The ATP is going to provide an energy source, and the reduced NADP is going to provide hydrogen. It's going to reduce other molecules, okay? So the last thing I want to finish up with on this topic, on this lesson within this topic, is let's review what we've seen so far in regards to the equation, that classic photosynthesis equation. Because it still holds true, it's still balanced the same from GCSE, but we can start to appreciate that, yeah, it's a lot more complicated. So we'll cut to that. Right, okay, so here's the equation, both the simple word equation and the balanced symbol equation. And hopefully you remember this from GCSE. I've certainly burned it into my brain over the years. And it's exactly the same, no matter whatever you look at it, it's correct. But obviously at GCSE you don't really understand how it works. So let's review. What have we seen so far from the light-dependent reaction? Well, we've seen water. We've seen that photolysis bit, haven't we? We've seen the oxygen that's been released or used in respiration. And we've also seen the light, haven't we? Okay, and the excitation of those electrons in the chlorophyll molecule. So we haven't seen carbon dioxide or glucose yet. No, they will appear in our next lesson, which is the light independent reaction. And we can start to account for some of these different elements in our glucose. So glucose is a carbohydrate because it's got carbon, hydrogen and oxygen in it, C6H12O6. And we can start to see that the hydrogen has come from the water, okay? The oxygen hasn't, though. So hopefully you've got an idea where the oxygen in our glucose has come from by looking at this. You probably get the idea. And we've now obviously isolated our oxygen, and that's been released there. So complicated stuff, difficult and challenging, I know, but you'll get there. As long as you work at it, you'll be all right. This is old Dr. P signing off. Stay safe. Have a good time. I think it's the weekend now, so enjoy. See you later.